Ever wondered how scientists tackle the mind-boggling complexity of cancer? It's not just one thing gone wrong. It's a tangled web of genetic, molecular, and cellular changes. But here's the exciting part. Researchers are now using cutting-edge tools to untangle this mess and find better ways to diagnose, treat, and even prevent cancer. Let's break down how they're doing it. Traditional approaches often look at one piece of the puzzle at a time, like studying DNA mutations or protein levels separately. But cancer doesn't work that way. It's a multi-layered problem. That's where multi-omics comes in. Think of it as combining different types of biological data, genetics, proteins, metabolites, and more, into one big picture. It's like solving a mystery by gathering clues from every corner of the crime scene. But here's the catch. These datasets are massive and interconnected. How do you even start making sense of them? Enter graph neural networks, a type of AI that's revolutionizing how we analyze complex systems. Imagine a web where each node is a gene, protein, or patient, and the connections between them reveal hidden patterns. These networks don't just process data, they learn the relationships like a detective spotting links between suspects. Take MOGONET, for example. It uses graph convolutional networks to map out how different genes interact in cancer. By doing this, researchers can classify cancer subtypes more accurately and even spot new biomarkers, like SFRP4, which we mentioned earlier. This protein might hold the key to predicting how well a patient responds to immunotherapy. Another tool, MOGAT, uses graph attention to focus on the most critical connections, much like how you'd zero in on the most suspicious clue in a mystery. These methods aren't just theoretical. They're already helping scientists uncover patterns that traditional tools miss. For instance, studies like DeepMoIC and MOGAT use deep learning to integrate incomplete data, making it possible to analyze real-world datasets that are often messy and incomplete. It's like cleaning up a cluttered room. Step by step, you start to see the furniture again. Let's get specific. Multi-omics data is inherently high-dimensional, with each omics layer, genomics, transcriptomics, proteomics, providing a unique perspective. But integrating these layers is like trying to read a book written in multiple languages. Graph neural networks, GNNs, solve this by constructing modality-specific graphs for each data type. For example, a DNA methylation graph might highlight regulatory regions, while a transcriptomics graph emphasizes gene expression patterns. These graphs are then fed into GNNs, which use message-passing algorithms to propagate information across nodes, identifying hidden interactions. The goal? To move from one-size-fits-all treatments to personalized medicine. By understanding the unique molecular fingerprint of each tumor, doctors could tailor therapies to individual patients. Imagine a future where cancer isn't just treated, but predicted and prevented with precision. Of course, this is still early days. But the tools are getting smarter, the data is growing, and the collaboration between AI and biology is accelerating. So the next time you hear about breakthroughs in cancer research, remember, it's not just about finding a single gene or drug. It's about connecting the dots in a way we've never been able to before. And that's pretty amazing. Thanks for watching. If you want to dive deeper into how AI is changing medicine, hit subscribe. The future of science is wild, and we're just getting started.